Hello boys, welcome back. Welcome back to another session of video tutorials, and this session is meant for class 12th biology students, and this is for 30th September 2020. So we are in the revision and we are dealing with chapter number 4, that is the reproductive health, and we have been discussing about the the biggest problem for some of the couples, that is infertility, and these unfortunate couples who were having the feeling that they have been cursed. Now our biology has shown a way that it will be no more a curse. Through biology there are so many techniques that can be used so that the issue-less couple will have children and they will be very, very happy. So basically infertility is the couples which are unable to reproduce children in spite of unprotected sexual cohabitation are called infertile. So it is the inability. It is caused by various reasons which can be grouped under physical, congenital, immunological, or even psychological disorders. So to overcome these issues, we have got a technique called ART, Assisted Reproductive Technologies. So this ART includes a number of special techniques which assist infertile couples to have children. So some of the important techniques, let us see now. In vitro fertilization, first one, IVF. So all these abbreviations in this chapter, what is more important is, you always in CBSE exam, the questions will be on abbreviations. Expand the abbreviations like IVF, in vitro fertilization. The baby produced by conceiving a culture dish and nursing in the uterus is called test tube baby. Of course, all of you know this IVF commonly is known as test tube baby technique. Test tube baby program. Okay, and this method involves IVF, in vitro fertilization. So fertilization of male and female gamete outside the body in almost similar conditions as that in the body, followed by ET. What is ET? Embryo transfer. Embryo up to 8 blastome years, you must remember this, 8 blastome years, that's the condition, is transferred into fallopian tube, and we call this technique as ZIFT. What is that ZIFT? Z for zygote, I for intra, F for fallopian, T for transfer. So ZIFT is zygote, intra, fallopian transfer. To complete its further development, that is from 8 blastome years, it keeps on increasing the numbers and stages will go on. If the embryo is with more than 8 blastome years, then it is transferred into uterus. Then we say IUT. If it is more than 8, below 8, ZIFT. If it is more than 8, IUT, intrauterine transfer into the uterus for complete development. A developing embryo can be inserted in the uterus of another female. So it is not necessarily that the same biological mother should bear this. So nowadays we have got volunteers from lady volunteers who can carry this. So they are called surrogate mothers. So the uterus will be given on rent. So these mothers are called surrogate mothers, not biological mothers. In case of some problem with the biological mother, the surrogate mother can have this embryo within her uterus and help her to complete the further development and give birth to the baby. So a woman who substitutes or takes the place of the biological mother to nurse the embryo in her uterus is called surrogate mother or genetic mother. So there will be two mothers. One is the biological mother and another one is surrogate mother or genetic mother. So you can call her more appropriately surrogate mother instead of genetic, I feel, okay, because she is not going to exchange any genes there, no, as such, because the ovum is not from her. It is only that she is giving uterus for rent, okay. And then second technique is GIFT. There it is ZIFT, remember, zygote intrafallopian. Here it is GIFT, gamete intrafallopian transfer. So transfer of gametes collected from the donor into fallopian tube of another female who do not produce ovum. Here see, the biological thing only, genes only will change, because the ovum, functional ovum, which we were talking about during ovulation and oogenesis, somehow in some ladies, unfortunately, that functional ovum also cannot be produced. So in such a case, the couple can ask for donors to donate their ovum. So when they donate, and that donated ovum can be transferred. So transfer of gametes, that's why we are saying transfer of gametes collected from a donor. So here the biological mother is unable to produce even the functional ovum. So for that also we have got a solution. And the third one, Intra Cytoplasmic Sperm Injection, ICSI, specialized procedure to form an embryo in laboratory in which sperm is directly injected. Look at this, sperm is directly injected into the ovum. So that is ICSI. So this is the technique where sperm is directly injected into the ovum to form an embryo in the laboratory. The embryo is later transferred by either ZIFT or IUT in women. So again, we have got this. Here if the problem is from the male, sperm is directly injected. Then we come to the next one, artificial insemination. It's called AIT, artificial insemination technique. So this is called, abbreviated as AIT, artificial insemination technique, where the husband is either unable to inseminate the female or has very low sperm counts in the ejaculation. This technique is used in those females. In this technique, the semen collected either from the husband or a healthy donor is artificially introduced into the vagina or into the uterus. That is intrauterine insemination, IUI. Intrauterine, another I, insemination of the female. Here the problem is with the male. Somehow the male is unable to produce sperms, and even if he produces the sufficient amount of sperms, mobility may not be there, and sometimes the number of sperms also will be less. In such a case, this artificial insemination, AIT, will be of great use. And then all these techniques, like whatever we have listed, that test tube baby program, that is IVF, in vitro, artificial insemination technique, gamete intrafallopian transfer, GIFT, and then ICSI, intracytoplasmic sperm injection. All these techniques require extremely high precision handling by specialized professional and expensive instruments. Therefore, their benefits are limited to certain countries and to certain groups of people. So emotional religious and social factors also prevent adaptation of these methods. Of course, biologically we are ready to help, but then what to do with this emotional and religious and social factors? Somebody, a lady, is she ready to receive a sperm from another male other than her husband? So these are the issues. Similarly, if there are some problems with religion, so these are the factors that will be always hindering whatever advances that we are doing in science. Anyway, it is left to the individual choice. There is no force in this, and we biologists are here to help. That's all. And we go to the next one, that is detection of fetal disorders. That is, in case the growing baby, by using all this, we ensure that the baby grows in the mother. And once the baby is growing inside the mother, is the baby growing normally? So to detect whether the baby is growing normal or in abnormal way, any defects in that, so we have some techniques. So trans-abdominal aspiration of fluid from the amniotic sac of the fetus is called amniocentesis. It's a fetal sex determination and disorder test based on chromosomal pattern in the amniotic fluid surrounding the developing embryo. Already we discussed this amniocentesis. The positive side of this amniocentesis is to detect any fetal disorder, and so that remedial measures can be taken. But what's happening? Negative side of it is the fetal sex will be determined based on the number of chromosomes and the type of chromosomes. Like I said, in the amniotic fluid, if there is XX, we know that it is going to be the baby girl. If it is XY, if you observe XY chromosomes along with the 44, that's male. But what's happening in some of the parts of our country, the moment parents come to know that it is female, the female is getting aborted, female feticide, we say. So the negative side of it is amniocentesis is female feticide. And then second one, there is CVS technique. CVS, chorionic villi sampling technique. The physician inserts a narrow flexible tube through the mother's vagina and cervix into the uterus and withdraws a small amount of fetal tissue, that is chorionic villi, from the placenta. One of the widely used non-invasive technique to determine fetal condition is, of course, all of you know, ultrasound imaging. And then there is one more called fetoscopy, is another technique in which a needle thin tube containing a viewing scope is inserted into the uterus, giving the physician a direct view of the fetus. So by employing amniocentesis, CVS technique, ultrasound imaging, fetoscopy, we can understand whether the baby growing inside the uterus, is it healthy, is it growing normally, or is there any abnormality. So the physician can help us to detect the abnormalities and remedial measures can be taken. So these are the various techniques we use. So I'm sure, being a biology boy, you must be very proud of all these achievements, and I want you in future that you should be part of such inventions, right? Until then, take care. I'll be posting again MCQs. Answer that. That will increase your self-confidence. Get prepared. And remember that Vijayanagar House slogan, when going gets tough. I know COVID-19, it's a tough situation, but remember the Vijayanagar House motto, when going gets tough, the tough gets going. It's not only Vijayanagar House boys, all other house boys must be